Today we will learn the chapter 4, the structure and function of fibrous proteins. Chapter 4 is composed of 4 subjects starting from amino acid composition and organization of fibrous protein. And second one is keratins and fibroin. Finally, we will cover collagen. So in this slide, I will explain the first subject, amino acid composition and organization of the fibrous protein. What is very common classes of the fibrous protein? That one is starting from the fibroin. So fibroin is the example, the silk. And how about the alpha keratin? Alpha keratin is also main component of hair and wool and nail. How about collagen? Collagen is, we can easily see collagen under the skin and also teeth and bones. So table 4.1 summarizes the amino acid composition of 3 common classes of fibrous protein in the mole percent. Okay, let's start from the fibroin. So this is fibroin, and this is their component starting from glycine, alanine, serine. But what is very big difference in the fibroin compared to the alpha keratin and collagen? So if you see here, okay, there are glycine and alanine and serine. The percent is higher than other amino acid components. For example, cysteine is nearly 0% in the fibroin, and also methionine is also 0%, and the other amino acid is also the very low percentage. But you see glycine and alanine and serine, they are main component of the fibroin. So what is common characteristics of the glycine, alanine, and serine? The common characteristics is their side chain. The size of their side chain is very small. Okay, so we will see some detail in the next slide. Okay. How about keratin? Keratin is the main component of the hair, wool, nail. If you see here, their serine percentage is about 10.2, and glutamic acid is 12.1, and cysteine is 11.2. Okay, the main very big difference compared to the fibroin and collagen is the cysteine. If you see here in the fibroin, the percentage of the cysteine is 0%. How about collagen? Collagen is also, the percentage of cysteine is 0%. Okay, so the structure of the cysteine is in the R chain, there is the CH2 and the SH. Instead of H, there is a SH group is there. Okay. Okay. So this is the very big the characteristics of the alpha keratin. Higher percentage of the cysteine. So how about collagen? Collagen, if you see here, collagen is the glycine is 32.7%, and alanine is about 12%. And how about another how about here, proline? Proline is in the fibroin, the proline percentage is only 0.3%, but alpha keratin is 7.5%. How about in collagen? The proline is around 22.1%. They show the higher percentage. Okay, the collagen is they have characteristics, especially is their glycine percentage is higher, and also their proline percentage is also higher. Okay, this is glycine and this is proline. Proline is usually observed in the tongue region. Okay, in tongue region. Okay, so this is so we will see why collagen is they are enriched with glycine and the in the next slide. Okay, in the next slide. Okay, so in this slide we summarize the amino acid composition and organization of fibrous protein. Once again, fibroin, the main component is the glycine, alanine, serine. These three amino acids they have the very small side chain. How about keratin? Keratin, compared to the fibroin and collagen, they have the higher cysteine percentage. And the collagen is they have the higher glycine and higher proline percentage. So we will see why the fibrous protein has this kind of characteristics. I will explain why this fibrous protein this kind of characteristics in the later. Okay. So in this slide we will learn the second subject, keratin. So we will learn about we will learn the alpha keratins. So what is main characteristics of the alpha keratin? We already learned that the cysteine content is higher in the alpha keratin. So now we will learn the basic structure of the alpha keratins. So alpha keratin is composed of the two right-handed alpha helices. Two right-handed means that N is start from here, and C is the carboxyl group is ended here. Okay, so it's composed of two right-handed alpha helices, and two alpha helices twist together to form a coiled coil. There are two kinds of coiled coil. One is parallel coiled coil, means that each strand is for example, they are composed of two strands, but all of strands, the N terminal is start from the left, and C terminal is ended at the right. We call it as parallel coiled coil. How about anti-parallel coiled coil? Okay, one strand is for example, this strand is that the N terminal is start from the right side, but however the other strand is N strand is start from the left side. So this one we call it as anti-parallel coiled coil. So we can usually see the alpha keratin in the hair and the wool and the nail. So now in the next slide we will see some special characteristics of the alpha keratin. So alpha keratin is composed of the repeating unit. So we call it as a heptad repeat. Heptad means that there are 7 amino acids is there, and at that, for example, this is the heptad repeat means that A, B, C, D, E, F, G. At that also A, B, C, D, E, F, G, like this. And also one strand, this is the coiled coil structure. So one strand is A, B, C, D, E, F, G. At that A, B, C, D, E, F, G. Another strand is also A, B, C, D, E, F, G. A, B, C, D, E, F, G. So usually this is the coiled coil structure. The two alpha helices, they forms coiled coil structure. So when they form the coiled coil structure, the contact region. So usually the A and D is the contact region of the coiled coil structure. So usually in this A and D amino acids is composed of the hydrophobic residue. We call it as the leucine and isoleucine, valine, aline, which has the aliphatic side chains. Okay, so A and D is most of them is composed of the hydrophobic residue. So it means that this residue is also hydrophobic, and this residue is also hydrophobic. So interaction between neighboring hydrophobic species, these species forms the coiled coil structure. Because there is the interaction between hydrophobic species, it means that this interaction is not strong. Okay, so when they forms coiled coil structure, there is interaction is happen, but this interaction is not strong. Okay, so after that, so E and G location and E and G location. So usually in the E location, glutamate is there. So glutamate has so you have to memorize all of amino acid structure. If I call it as glutamate, you have to think about E. And also you have to think about they have the carboxyl group. How about G? So G and G. So G is usually composed of the arginine or lysine. Arginine they have the guanidinium group. They have positively charges, and also lysine. Finally they have the amino group. This is also positive charges. So between E and G, there is the charged interaction happens. So even though the interaction region is the hydrophobic interaction, but between E and G, charged interaction happen. So that's why the coiled coil structure, when they make coiled coil, they can be stable because of the charged interaction. So, okay, so in the E location, fifth location, usually they are composed of the glutamate and also G. G means that 1, 2, 3, 4, 5, 6, 7 amino acid is usually the arginine or lysine, which has the positively charged side chain. Okay, so this is the repeating unit of alpha keratin. Once again, heptad repeat. Okay, so you have to memorize which amino acid is located in A and D, and which amino acid is located in the E and G. So that's why between E and G, the charge interaction happens. So even though when they form the coiled coil, the contact region is the hydrophobic interaction. It's very weak interaction is there, but at that there is between E and G, there is the charge interaction is there. Okay. Alpha keratin showed that higher order structure, starting from the alpha helix. One strand is alpha helix. After that, two alpha helix twist together to form dimer. So two alpha helix twist together to form dimer. So they forms coiled coil structure, and after that their basic structure is they are composed of the head to tail order. Head is composed of the N-terminal globular domain, and the tail is composed of C-terminal globular domains. So they show that the head to tail structure. Okay. How about the protofilament? So how about protofilament? So this is protofilament. So usually the two dimer associate to form protofilaments. Two dimers associate to form the protofilaments. So they show that head to tail, 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 head to tail. They show that the head to tail order. Okay. So next one is that this is protofilament. Next one is the protofibril. So next one is protofibril. So this is protofibril and this is protofibril. Okay, protofibril means that two protofilaments associate to form protofibrils. Two protofilaments associate to form protofibrils. So after that, the final, the very complex one, we call it as the microfibril. So this is microfibril. 1, 2, 3, 4. 1, 2, 3, 4 protofibrils associate to form microfibrils. 1, 2, 3, 4. 4 protofibril associate to form microfibril. So the simplest one, starting from the alpha helix, after that it will make the two forms dimer. Two dimer associate to form protofilament. So both of them end to head to tail order, head to tail order. Except that two protofilaments associate to form protofibrils. Okay, after that 1, 2, 3, 4 protofibrils associate to form microfibrils. Okay, and also the basic subunit dimer, the size is about 450 angstroms. So usually if we so it means that this is 45 nanometer. Okay, 45, something like that. Okay, so this dimer is the basic subunit, and after that the complex one is microfibril. Okay, so in the previous slide we learned that two alpha helix forms cold coil structure, so we call it as a dimer, and two dimer associate to form protofilament, and two protofilament make the protofibril. After that 1, 2, 3, 4 protofibrils make microfibrils. So microfibril is main component of the intermediate filament. So in the cytoskeletal, so there are three types of the cytoskeletal filament. One is actin filament, and the other one is the microfilament. But microfibril is the main component of the intermediate filament. So in this slide, let's classify the intermediate filaments. There are several types of the intermediate filaments. So in this slide, we will cover type 1 and type 2. Type 1, we call it as the acidic keratin. Type 2, we call it as basic keratin. So type 1, so alpha keratin is their basic subunit is they have the heptad repeats A. So starting from the A, B, C, D, E, F. So A, B, C, D, E, F, G, like this. Okay. So A and D location, there are hydrophobic interaction is happen. But usually E and G location, Usually E location, the glutamate is there, and G location, lysine and arginine is there. So except for A, D, E, G, so there are B, B, C, there are another, the amino acid is here. So in these locations, so usually the type 1 acidic keratin, they have lots of the negatively charged side chain here, 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 here. So the negatively charged side chain means that glutamate E and aspartate D. So type 1 intermediate filaments, they have lots of the negatively charged side chain in this location. How about type 2? So type 2 is also, we call it as basic keratin. So basic keratin means that they have lots of positively charged side chains. So A and D location, so they have the hydrophobic residue is A and D location, and E and G, E and E location, they have glutamate, or usually they have the glutamate. But in the G location, they have the lysine and arginine. So between E and G, there is the ionic interaction happens. But except this location, so in this location, type 2 basic keratin, they have lots of positively charged side chains, the lysine or arginine. Okay. So table 4.2 summarizes the classification of intermediate filaments, starting from type 1, 2, 3, 4, 5, 6, and unclassified. Okay, but usually the most major form of the intermediate filament is type 1 and type 2. So usually the 90% or 95% of intermediate filament is type 1 or type 2. So let's see the location of type 1. So type 1 is acidic keratin. So their location is the epithelial. So epithelial means that is the 표피. In Korean we call it as 표피. Type 2 is the basic keratin, is also their location is epithelials. So type 2 is So type 2 is composed of from K1 to K8 intermediate filament, and type 1 is comprised of from K9, K10, and K21 intermediate filaments. Type 1 and type 2 keratins are distributed in different combination in different cells. So this is the human skin epidermis. The outer region is the comprised of epidermal keratinocytes, and usually in the middle region there is the supravasal keratinocytes, and the bottom region there are basal keratinocytes. For example, in the middle region, supravasal epidermal keratinocytes is usually composed of K10 and K1. How about basal keratinocyte? Basal keratinocyte usually composed of the K14 and the K5 keratinocyte. K5 intermediate filaments. K5 keratinocytes. K5 intermediate filaments. So in this slide, I will explain its interaction stabilize alpha keratin. The strongest one is disulfide bond. So disulfide bond happens between cysteine and cysteine, and also between cysteine and cysteine. So it happens between the adjacent alpha helix and also within the alpha helix. It happens. The reason why the disulfide bond easily happen in the alpha keratin, it means that the main component of alpha keratin, main amino acid component is cysteine. The average percentage is about 11.2%. Okay, so if the alpha keratin shows higher cysteine content, for example 15%, so it induced formation of harder alpha keratin, especially the nail. Nail has higher cysteine content compared to the wool or compared to the hair. Okay, second strongest one is ionic bond. Okay, between the positively charges. So E location, positively charged amino acid is here, and G location, usually the E location, the negatively charged carboxyl, and the G location, positively charged amino acid is here. So between the positively charged and negatively charged amino acid, the ionic bondage happen. And also hydrogen bondage happen between the strands, okay, or within the strands, within the alpha strands. Okay, and also final one, the weakest interaction is the hydrophobic interaction. So, okay, so hydrophobic interaction usually happen between A and D. So A and D is happen in the coiled region. Okay, okay. So in this slide we learned that the interaction stabilizing alpha keratin starting from disulfide bond and the ionic bond, hydrogen bond, and hydrophobic interactions. Okay. So in this slide we will learn the third subject, fibroin. So fibroin we call it as silk. So silk consists of antiparallel beta strand. So usually the antiparallel beta strand show that fully extended conformation. Do you know why? Because we learned that in alpha helix they are composed of 3.6 residue per turn. So per turn, so they progress 0.45 nanometer. How about the antiparallel beta strands? So this is the antiparallel beta strand. One is strand starting from N to C, the other N to C, N to C is like this. So each turn they are composed of the two residue. So especially the antiparallel beta strands, 0.68 nanometer is progressed per turns. Okay, okay. But how about the fully stretched polypeptide? So we learned that the distance, the separation distance between the amino acid and amino acid, one residue is about 0.38 nanometer. How about two residue? Two residue is 0.76 nanometer. So if you compare here and there, so we can say that the antiparallel beta strands, two residue per turn, they progress the 0.68 nanometer. So it means that antiparallel beta strand, they are nearly fully extended conformation. However, the alpha helix, this is not fully extended because one turn they are consist of 3.6 residue, but they only progress 0.54 nanometer. So, for example, alpha helix can be stretched, but antiparallel beta strand cannot be stretched. So silk cannot be further stretched without breaking covalent structure in beta strands. So without breaking covalent structure in beta strands, so silk cannot be further stretched. Okay, so it means that silk is very stable. Okay, so in this slide I will further characterize the fibroin structure. So fibroin is they have the composed of repeating unit, the glycine, serine, glycine, alanine, glycine, alanine. So they have this kind of hexapeptide repeats. So you see here glycine, serine, glycine, alanine, glycine, alanine. Okay, so they have the repeating unit. So where is the repeating unit existed? Okay, so this is a repeating unit. Okay, I will show you. So this is silk. Silk is composed of the fibroin. Fibroin is composed of fibroin fibrils. So if you magnify this region, so this is the silk. So they are composed of the antiparallel beta strands. So each antiparallel beta strand is connected by gamma strand. Okay, so each beta strand is they show that hydrophobic. In gamma strand is they are hydrophilic. Okay, this is hydrophilic region. Okay, so once again, if you magnify this region, so the antiparallel beta strand is hydrophobic, and the connecting gamma strand is hydrophilic domains. So each hydrophobic domain is composed of the hexapeptides. Okay, so between the hexapeptides there are tetrapeptides existed, but we will not cover in this slide. Okay, so main repeating unit is hexapeptides. So hexapeptide is composed of glycine, serine, alanine. So you can see that the side chain is the very smallest, isn't it? Okay, and also each antiparallel beta strands. The main component of each antiparallel beta strand is the glycine, serine, glycine, alanine, glycine, alanine. So this region is the hydrophobic region. Hydrophobic, okay, because they don't have any charge. Okay, hydrophobic region. So between this region, usually the van der Waals interaction happens. So between the hydrophobic region, the van der Waals interaction existed between antiparallel strands. So because the weak van der Waals interaction existed between antiparallel beta strands, so that's why silk is very flexible. Silk is very flexible. The reason because the weak van der Waals interaction exists between antiparallel beta strands. The important thing is that repeating unit. So we have to memorize repeating unit of silk, the glycine, serine, glycine, alanine, glycine, alanine. Okay, this is the repeating unit. Silk has the hexapeptide repeat, which is composed of glycine, the alanine, and serine. So these three amino acids is the amino acids with the smallest side chain. This amino acid consists of the 85% in silk. It means that it can induce close packing of antiparallel strands. Okay, so this is one antiparallel strand, this is the other, and this is the other, something like that. Okay, so between one strand and the other strands, there is the interaction of the alanine or serine residue in antiparallel beta strands. Next to this strand, okay, there is in this strand there is only glycine residue existed between these antiparallel beta strands. Okay, it means that the interaction of alanine and serine residue in this region is the packing strength is about 0.57 nanometer. But in this region, the interaction in this region, there is only glycine side chain is existed in this region. Between this antiparallel beta strand and this antiparallel beta strands, there is the only glycine residue existed. So interaction of glycine residue in this region, they show that they have 0.35 nanometer. So it means that we can closely pack antiparallel strands. So it means that it can induce the silk is very strong characteristics. Okay, even though they are flexible, so they have the very strong characteristics. Okay. So in this slide, I will explain the fourth subject, collagen. So table 4.4 summarizes the major collagen group starting from type 1, type 2, type 3, and type 4. So type 1 is the most abundant the collagen, and type 2 is second abundant collagen. Okay. Type 1 is the most abundant collagen, is the chief component of skin, tendon, ligaments, and bone. Okay, if you see here, so this picture comes from Nature Chemical Biology journal. And also if you see here, so this is mouse skin. So red area is mouse epidermis. So below the epidermis, there is collagen is there. So collagen is located below the epidermis. Okay, below the epidermis. Okay, so this is the collagen. The blue area is collagen. So this picture comes from Nature Chemical Biology. So who published this paper? It's me. I published this paper in Nature Chemical Biology. So I stained the mouse epidermis, mouse skin, and found that the collagen was deposited like this, okay, in blue color, okay. And also we can easily see the type 1 collagen in 돼지 껍데기, okay. And also here is this is tendon. So tendon is bound, so this is tendon, and this is muscle, and this is bone. So tendon is bound muscle to bone. Okay, in Korean we call it as 힘줄, and ligament means that we call it as inde. This one is bound bone to bone. Okay, after there is the bone is also major component of bone is also collagen. Okay, so type 2 is the second abundant collagen, and also the chief component of cartilage. So this is the cartilage. So the cartilage is composed of the type 2 collagen. So we call it as a 연골. Okay. So and also there are type 3 and type 4 is not main component. So today's lecture I will not cover. So we cover type 1 and type 2. Type 1 is most abundant and also chief component of skin, tendon, ligaments, and bones. Type 2 is the second abundant collagen and also chief component of cartilage. In Korean, we call it as the 연골. Okay. So in this slide, I will explain the collagen structure. So collagen is composed of 3 left-handed helix. Okay, each they have the 3, 1, 2, 3. Each one is left-handed helices. Okay, the bottom is that N-terminal and above is C-terminal, so they are left-handed. So each helix is left-handed. Okay, how about alpha keratin? So alpha keratin is composed of 2 right-handed alpha helices, but collagen is composed of 3 left-handed helices. So these 3 left-handed helices finally form triple helix, but this triple helix is right-handed. Each subunit, each helix structure is left-handed, but their final form is they will make the right-handed triple helix. And also, collagen is also composed of a basic subunit. We call it as they have a repeating unit. Okay, one is a glycine, XAA, YAA. So XAA means that proline and lysine can be here. YAA means that proline and lysine can be located here. Okay, so it means that the repeating unit will be the glycine, proline, or glycine, proline, lysine, or glycine, lysine, proline, or glycine, lysine. Okay, so because they have the proline and lysine as a repeating unit, so usually the post-translational modification is happen. For example, proline. So this is proline. Proline has the C alpha, C beta, C gamma carbons. So usually the C gamma carbon is the methylene, means that they have CH2, but instead of the H, OH is bind to this C gamma carbon. So this one we call it as hydroxyl proline. How about the lysine? So this is a lysine structure. So lysine, we have the C alpha, C beta, C gamma, C delta. Initially, the C delta carbon is methylene CH2, but instead of H, OH group is here. So we call it, this is hydroxyl lysine. So hydroxyl proline, we write down HYP, and hydroxyl lysine, we write down HYL. So we will see the effect of the hydroxylation in the collagen. We will see the effect of post-translational modification in the later slide. Okay. So you have to think about what will be the effect of the post-translational modification in the collagen. Okay, so this is the collagen structure. The important thing is each subunit is left-handed helices, but they will make 3 left-handed helices from right-handed. This is a right-handed triple helix. Okay, and they have a repeating unit, glycine, XAA, and YAA. Okay, so and also previously we learned that alpha helix is composed of the each pitch. So each turn they are composed of 3.6 amino acid residue, and each turn they progress 0.45 nanometer. So actually the translation distance per residue is 0.15 nanometer. Okay, so this is alpha helix. How about collagen? Collagen is a left-handed helices. Okay, so each turn they are composed of 1 amino acid glycine, for example, proline, and hydroxyproline. After that, they have 0.3 amino acid. For each pitch, so the collagen helix has a pitch in angstrom. They have they are composed of each pitch or each turn. They are composed of 3.3 amino acid residue, and they progressed 0.94 nanometer per turn. So it means that angstrom means that 9.4 angstrom. Okay. The big difference between the alpha helix and the collagen, the left-handed helixes, is that, for example, in alpha helix, the translational distance per residue is 0.15 nanometer. However, in the collagen, the translational distance per residue is 0.286 nanometer. So it means that each residue, they progress is more. Okay. So when you compare the stability between the alpha helix and the collagen, which is more stable? When you compare the stability between alpha helix and the left-handed the helixes in collagen, so alpha helix is more stable than the collagen helix. Okay, because they progressed, the translational distance is the 0.15 nanometer, but this one is about 0.286 nanometer. So it means that this collagen structure will be more unstable than the alpha helix. Okay. So in previous chapter, we learned that alpha helix structure. So alpha helix, so each pitch, each turn, they are composed of 3.6 amino acid residue, and each turn they progress 0.45 nanometer. So how about the collagen? Collagen is the left-handed helix. Okay, let's start from here. Glycine, proline, hydroxyproline. After that, there is glycine. To make each full pitch or each full turn, one turn, they are composed of 1, 2, 3, and 0.3 amino acid. Okay, so it means that per turn, they are composed of 3.3 amino acid residue. And each turn, they progressed 0.94 nanometer. Okay, so it means that what is collagen helix has a pitch in angstrom, means that 9.4 angstrom. Okay, so it means that the translational distance per residue is 0.286 nanometer. So when we compare to collagen structure and the alpha helix, so based on the translational distance, in this alpha helix, the one residue, they progressed 0.15 nanometer. For example, in collagen, they progressed 0.286 nanometer. It means that nearly two times larger than the alpha helix. The collagen helix is 2 times larger than the alpha helix. Okay, it means that when you compare to alpha helix and collagen helix, it means that alpha helix is more condensed, more compact than the collagen helix. It means that alpha helix they are more compact. It means that they are more stable. Collagen is a little bit unstable. Okay. So collagen is their repeating unit is composed of the glycine and XAA and YAA. So XAA, there will be the proline can be there, and the YAA, and also proline can be there. Okay, so this is the regular polypeptides. So they are connected by each amino acid is connected by their peptide bonds. For example, here they are connected by peptide bond, peptide bond, peptide bond. Okay, so NH. So in these cases, NH, they have NH. So this usually this amide hydrogen, they make the hydrogen bonding between the neighboring the oxygen. Okay. However, if you see your proline, so this is proline. So when they make the polypeptides, if there is proline, they change it. They make turn. Okay. If you see here, they only have N. For example, the other amino acids, they have NH, but however, the proline, they only have N. So it means that absence of amide hydrogen, so H, the NH, the hydrogen eliminates any potential hydrogen binding. So it means that weak interaction with the neighboring amino acids. So usually when they make the triple helix, they make the hydrogen bonding with the neighboring amino acid. But however, the main component of collagen is the proline. But proline don't have the amide hydrogen. They don't have H here. So this H, absence of this H hydrogen eliminates any potential hydrogen binding. So it means that weak interaction with neighboring amino acids, it induced weak interaction with the neighboring amino acids. So it means that this triple helix is very weak compared to the alpha keratin. Okay, how about they also have another basic subunit, the glycine. They have the glycine, XAA and YAA. So glycine occurs every third residue. So usually this is triple helix. So usually glycine is located near the helix axis. So glycine always occurs here, 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 here. So glycine occurs every third residue and locates near the helix axis. So it means that glycine has minimal side chain. So this is glycine. They have a minimal side chain. They only have hydrogen as the side chain. So this minimal side chains allows the formation of triple helix. So they can make because the glycine don't have the side chain, they have a minimal side chain. So this minimal side chain can allow formation of triple helixes. So if for example, in the glycine location, if the tryptophan has very big, large side chains, so if the tryptophan was there, so it will be very difficult to form triple helixes. But usually the glycine occurs every third residue, so it also locates near the helix axis. So it means that glycine has minimal side chain. So minimal side chains allows to form triple helixes very easily. Okay. So in this slide, we will learn the interaction stabilizing collagen structure. Previously, we learned that interaction stabilizing alpha keratin and also interaction stabilizing fibroin. Okay, first of all, in the alpha keratin, so there was the strong disulfide bond was happens between cysteine residue, and also there was the ionic bond, and also hydrogen bond, and finally there was a hydrophobic interaction between the A and D region. Between A and D region, there are the amino acid was there, especially the leucine or isoleucine or valine or the alanine, which has aliphatic side chains. So that's why the hydrophobic interactions happens. Okay, how about in the fibroin? So there was the van der Waals interaction happens between the antiparallel beta strands. Okay, so usually the van der Waals interaction happens between the hydrophobic region. Okay, so this region was hydrophobic region. Especially in collagen structure, in the near the helix axis, there was only the glycine residues. So between the glycine residue, the van der Waals interactions happens among glycine residue near helix axis. And also hydrogen bonding happens between the amide hydrogen in the glycine and also between the carbonyl oxygen, this is carbonyl oxygen of XAA residue. The repeating unit of the collagen is glycine, XAA and YAA. So in XAA, so the hydrogen bonding happens between the glycine and XAA, especially the amide hydrogen in the glycine and carbonyl oxygen of the XAA residue. Okay. The third interaction stabilizing collagen structure is a covalent bond. Okay. So the repeating unit of collagen is glycine and XAA and YAA. In the XAA location, the proline and lysine can be there. And also YAA location, proline or lysine can be there. So it means that lysine is main component of the collagen. So this is the lysine structure. So let's see the detailed structure. They have alpha carbon, beta carbon, gamma carbon, delta carbon, epsilon carbon. So they also have two amino group. This amino group we call it as epsilon amino group, and this amino group we call it as alpha amino group. So usually the lysyl oxidase induced oxidation of the immune epsilon amino group of lysine. So it will oxidize the epsilon amino group of the lysine, so to forms aldehyde, carbonyl COH. So this we call it the aldehyde. So finally this full form we call it as the lysine. Actually this was a lysine. So the amino group, epsilon amino group, was the oxidized to form aldehydes. So we call it as this is a lysine. So condensation reaction happened between two lysines. So we call it as the lysine aldol condensation. So I will not cover detail in the aldol condensation, but important thing is that the covalent bonding is happen, especially the aldol condensation. So it means that their bonding is very strong. They make the very strong covalent cross links. So this covalent bond can strongly stabilize the triple helixes. Okay, but previously we learned that the van der Waals interaction and hydrogen bonding is a very weak interaction, but this covalent bonding is very strong interaction. So this due to this covalent bonding, triple helixes can be very strongly stabilized. Okay. So in this slide, I will explain the importance of the hydrogen bonding. So the importance of hydrogen bonding to stabilize the collagen structure. Okay. So this experiment, we did it like this. So we made the collagen structure which has the basic repeating subunit composed of glycine, proline, proline, and glycine, proline, proline. And also we also have another group which has a glycine, proline. Instead of the proline, we make the hydroxyl proline. So in these cases, glycine, proline, hydroxyl proline, and glycine, proline, hydroxyl proline is there. Okay. So usually the post-translational modification can be happen in proline or lysine, especially the proline. So this is a C alpha, C beta, C gamma carbon. So usually the C gamma carbon they have the methylene group, but now is the hydrogen is changed to hydroxy. So this is we call it as a hydroxyproline. Okay. So after that we measure the transition midpoint temperature. Okay. So we measure Tm. So in the case of the glycine, proline cases, Tm is around 17 centigrade. However, in the glycine, proline, hydroproline case, this case we measure the Tm's around 37, nearly 2 times higher than the previous one. So it means that this hydroxylation induced the triple helix stabilization. So triple helix can be stabilized due to hydrogen bonding between the hydroxyl proline and the neighboring chains. So usually the hydrogen bonding happens between the hydroxyl proline and neighboring chains happen. So it means that it can induce triple helix stabilization. That's why the glycine, proline, hydroproline, the Tm, transition midpoint temperature, is 2 times higher than the glycine, proline cases. Okay. So in this slide, I will show another example to show the importance of the hydrogen bonding to stabilize the collagen structure. So we can go over the hydrogen bonding in collagen again. So usually the hydrogen bonding happens between the amide hydrogen in the glycine and also carbonyl oxygen in the XAA residue. So usually the main component of gelatin is the collagen. Okay, when collagen was heated up, so hydrogen bonding between the triple helixes is broken. So it will make the disordered status. So it will make the Joule status, the liquid status. So when we cool it down, the gelatin, so it will regenerate triple helixes between the neighboring strands. So it will make the gel status. So gelatin status, gelatins, if we see the details, the gelatin, the hydrogen bonding is randomly hydrogenized. Hydrogen bonding is happen randomly in the neighboring strands, so it will make the gel status. Okay, so this experiment also showed that the importance of hydrogen bonding to stabilize the collagen structure. Okay. So in this slide, I will explain the collagen biosynthesis procedure, especially focus on post-translational modification. Because step 1 is that pre-pro-collagen was synthesized on the ribosome. So this is mRNA, and pre-pro-collagen, which contains the signal peptides, is synthesized on the ribosome. After that, pre-pro-collagen was transported to the lumen, the ER. So after that, the signal peptides of the pre-pro-collagen was cleaved in the lumen of the ER. So the first post-translational modification is cleavage of signal peptide or cleavage of signal sequence in the lumen of the ER. So this is first post-translational modification. So after that, step 2, so this is the collagen precursor. So with the N-terminal and N and C-terminal extension. So after that, step 3 is the second post-translational modification happens, which call it as a hydroxylation. So usually the hydroxylation happens in the proline and the lysine. We learned that the specific site in the proline and the lysine, okay, hydroxylase happen. After that, step 4 is that asparagine-linked oligosaccharides is added to the collagen. So especially the asparagine amino acids. So based on the asparagine amino acid, there are several oligosaccharides can be added. Okay, so step 4 is that the addition of asparagine. So at the base there is asparagine amino acid. So after that, there is oligosaccharides can be added. So addition of asparagine-linked oligosaccharides to collagen. So this is a third post-translational modification. After that, step 5 is that the hydroxylated region, especially the hydroxylysine. So there are two sites, hydroxylated, hydroxylproline and hydroxylysine. So especially the hydroxylysine is glycosylation is happen. So glucose can be added to hydroxylysine residue. So this is a post-translational modification. Step 5, during step 5, glycylation of hydroxylysine residue. Okay, and step 6 is that alignment of 3, 1, 2, 3 polypeptide chain, and also the 5th post-translational modification is that disulfide bond formation between the cysteine residue. So disulfide bond formation is 5th post-translational modification. After step 7 is that formation of triple helical structure. So we call it as the procollagen. So this is, we call it as procollagen. So step 7 is the formation of triple helical procollagen. Okay, this is, we call it as procollagen. Okay. So after that, triple helix was transferred to the transport vehicle. This procedure we call it as endocytosis. After that transfer, triple helix can be transferred to the extracellular region. So this process we call it as exocytosis. After that, this triple helix has the N-terminal peptides and also C-terminal peptides. So step 10 is the sixth post-translational modification. It means that it will remove N and C terminal peptides by specific peptidides, except that it will make the tropocollagen. So tropocollagen synthesis is happen by removal of the N and C terminal peptides. Okay, so this is the sixth post-translational modification. After that, each tropocollagen, this is each tropocollagen, so lateral association of tropocollagen is happens. So this one is between each tropocollagen covalent bonding is happens between these two tropocollagen, and also these two tropocollagen also covalent bonding is happened. So covalent bonding is happened. The covalent bonding is happened. So finally, fibril was synthesized. Okay. So the seventh post-translational modification is the covalent bonding between the tropocollagen. So this covalent bonding is induced lateral association of tropocollagen. So finally, fibril is synthesized. So from the step 1, 2, 3, 4, 5, 6, and from step 1 to step 11, we observe the seventh post-translational modification. Except that finally, fibril means that this is the collagen. Collagen is synthesized. Okay. So in this slide, I will explain the disease associated with collagen defects. The first one is osteogenesis imperfect. In Korean, we call it as the 골형성 부전증. The main cause of the osteogenesis imperfect is that single base mutation. So we call it a missense mutation in nucleotide sequence encoding the glycine. So this is the nucleotide sequence encoding the glycine. So single base mutation happens. So missense mutation happens. So this mutation substitutes glycine for another amino acid. So this substitution destroys the repeating unit. The repeating unit is composed of glycine and XAA and YAA. So what's the function of glycine in the collagen? So the glycine, we already learned that glycine occurs every third residue. So this glycine is located near the helix axis. So because they have the minimal side chains, it can easily allows the formation of the triple helix. So it means that if the repeating unit is destroyed, so it means that this triple helix cannot be easily made. So it means that it will induce several the results. First one is that they will induce imperfect bond formation. So it will induce the short status, okay, like this. There are several types of the imperfect osteogenesis imperfect, type 1, type 2, type 3, type 4. Nearly all of this type, they show the short status like this, okay? And also this is the bone structure. This is a healthy person's bone structure. But this bone, the patient which has osteogenesis, you will see that they have a lot of the blank spaces in the bone. So it will make the brittle bone. So it means that bone is fragile very easily. Okay, so this disease is due to the single base mutation in nucleotide sequence encoding glycine. encoding glycine. That's why it destroys the repeating unit, especially the glycine. So it means that usually the glycine has minimal side chains, so it can allow the formation of triple helix very easily. But in the case when the tryptophan is located in this location, so it means that it cannot easily make the triple helixes. So the consequence is it will induce imperfect bone formation, and also so bone is fragile is very easily, okay, like this, because the bone has a lot of space inside, okay. But normal healthy person has the bone is very densely packed, but this disease has brittle bone, okay. So next disease associated with collagen defect is the Ehlers-Danlos syndrome. The cause is the same. The cause is the single base mutation. Usually the missense mutation happens in the nucleotide sequence encoding glycine. So it substitutes glycine for another amino acids. So this substitution destroys the repeating unit. So as I mentioned earlier, glycine occurs every third residue. It locates near the helix axis. So because they have the minimal side chains, it allows formation of the triple helixes like this. But so that destroys this disease. The consequence is that they show that hyper-extensible joint, and also their skin is very soft, and they are elastic skins like this. So this is the disease which is associated with collagen defect. The first one was osteogenesis imperfect. The cause is also the single base mutation or missense mutation in nucleotide sequence encoding glycine. And also the second disease is the Ehlers-Danlos syndrome. They have same cause, and also it destroys the repeating unit, especially glycine. So the consequence is the hyper extensible joint and soft and elastic skins. Today we learned the amino acid composition and organization of fibrous protein. We also learned especially the keratin and the fibroin and collagen. Okay, so today we will finish the class.